correct.
Okay, great. Yeah, as mentioned, I'm a postdoc in Fritz Ross Lab, now at the University of Pittsburgh, but most of this work was done at the University of Toronto. And pretty much what we did was a mutational scan for serine threonine kinase 11, or STK11. So first off, I'll put serine threonine kinase 11, STK11, into the context of how it fits into the variant interpretation crisis, and then transition into talking about its clinical relevance both in a rare monogenic disease, but also in somatic cancers as well. For the latter half of the talk, I'll focus on the mutational scan that we performed to characterize thousands of different variants simultaneously. And really, one of the most interesting parts in my eyes, it will be integrating that data, the results that we have with phenotypic data for really the purposes of improving patient stratification and, of course, to help with variant classification. So here, what I've done is I've pulled data from ClinVar, so a database of clinical variation, and on the y-axis, I'm showing the number of unique missense variants that have been seen in STK11, and the x-axis is years over the past decade. And so what should really jump out is that variants that have been classified as pathogenic or benign have remained in relatively equal proportions over the past decade, but really it's that variants of uncertain significance that have continued progressively to increase over time. There are almost 1,000 different unique missense variants of uncertain significance in STK11, and really what we're hoping to do is to address this problem and invert this curve to provide new evidence for these variants of uncertain significance and hopefully help reclassify them towards pathogenicity or benignity. So STK11 somatically can be found in a variety of different cancers, but most commonly up to 10 to 30% of lung cancers and 10 to 20% of cervical cancers. But there are also several other tissue types it affects, but at lower frequencies. On the germline side, it results in Peutz-Jeghers syndrome, or PJS. And this is a rare monogenic disease that confers both a gastrointestinal complications, but also a multi-organ increased risk of developing cancer. So both on a germline and somatic side, there is an increased risk of cancer. So first focusing on somatic cancers and specifically looking at lung cancers. So one of the resistance mechanisms that lung tumors employ is expression of programmed death ligand 1, PDL1. And typically what happens is when a cytotoxic T cell comes to clear the tumor, binding of PDL1 to its receptor on these T cells results in anergy or apoptosis, preventing it from functioning. Modern immunotherapies serve to block this interaction by providing antibodies to either the ligand or the receptor, and so that these T cells can function, they can clear the tumor, and the patients are likely to have a better outcome. Where STK11 comes into play is that loss of STK11 reduces immune cell recruitment to the tumor, and so we have what's been described as a cold or inert tumor microenvironment. And so providing immunotherapies has limited benefits for these patients because the T cells simply aren't present. And this is a case where these individuals likely would benefit from an alternative therapeutic rather than immunotherapy. On the germline side of things, for Peutz-Jeghers syndrome, there are several indicators for a diagnosis, one of them being mucocutaneous hyperpigmentation, this being dark spotting on the lips, ears, or eyes. Of course, it's a germline condition, so family history. If you have a relative, a sibling, a parent that has the condition, then you're, of course, potentially at risk. One of the hallmarks of the disease are intestinal polyps. And so one method of diagnosis is to perform a relatively invasive procedure, a colonoscopy and endoscopy, and to observe these polyps. And that can be definitive, but it is somewhat invasive. And most relevant for my project is that since this is a germline condition, a simple blood test can potentially identify an STK11 variant and help with a diagnosis. But really, at this point in time, as mentioned previously, the vast majority of variants in STK11 are VUS, and so this really complicates this as a viable method for diagnosis. So here, just as a TOR example, let's say a patient comes in, has a tumor biopsy sequence, maybe they have a blood test. We identify the variant by sequencing, but there isn't that contextual additional information available to enable clinicians to act accordingly. So what we're hoping to do is to proactively assess nearly all substitutions in an experiment-based system. And so now immediately upon sequencing, we have a much richer phenotypic output, and the clinician can hopefully act accordingly based on the data that is already available. So really, the project that I'm talking about is really centered around generating a variant effect map for STK11. So now I'll transition to the latter side, which is focusing on really the deep mutational scanning aspects of this project. So first off, there is, of course, a whole pipeline that can be described, but every lab has their own methodologies. Generally speaking, you can split it into generating the mutagenic libraries, cloning into appropriate vectors, transfecting or transforming into the cell model of choice. Here we are using HeLa cells, so mammalian cells. The selection, which I'll talk about next slide, in this case is a growth-based assay. And finally, we sequence both before and after selection to compare the frequency of variance in the population and translate that into functional scores to generate the full variant effect map. So for the selection, essentially what we've done is we've reintroduced STK11 to HeLa cells, which normally do not express STK11. And so reintroduction reduces their proliferation rate because STK11 is a tumor suppressor. And so in a population where each cell receives a single variant of interest, over time, variants that are tolerated, that retain the protein's function, are going to drop out of the population, while variants that are damaging, that are pathogenic leaning, are going to take over the population. And this can be translated into frequency of variants by sequencing by doing a tiled approach of tiled primers across STK11. And so early time points, there should be relatively equal proportions of pathogenic, benign, and VUS, while at the later time points, really the pathogenic variants should be predominant in the sequencing reads, and there should be a very few number of benign variants that are left in the population. So this is the assay that underlines. It's a growth-based assay of expressing to STK11 in HeLa cells. So one of the internal quality control metrics that we always look at is, of course, synonymous and nonsense. Here, synonymous is in green, nonsense in blue, and I'm showing a moving window across the positions of STK11, with functional scores being on the y-axis. And so generally speaking, for most of the protein, from about positions 22 to 311, there is good separation between synonymous and nonsense, where synonymous have scores near 1 and nonsense have scores near 0. But what should really jump out is both the C-terminus and potentially the N-terminus here as well. For the C-terminus, this actually aligns fairly well with the end of the kinase domain at position 309, and it is well established that for certain proteins, a portion of the C-terminus is dispensable for function. But what is striking, is, of course, is the sheer number of amino acids. So this is nearly 100 or over 100 positions that seem to be dispensable for activity, for function of this protein. One very interesting finding from my eyes is sort of this tolerated section in the N-terminus here, up to about position 22 or 21. And initially this was very puzzling, but we believe that this could be indicative of an alternative translation initiation site where position 21, there's a methionine that matches the ribosome recognition motif, and this could potentially circumvent the early nonsense variants and provide them with a still functional protein. So if we look at this data in another way, specifically looking at the position 22 to 311, where we expect to see separation between nonsense and synonymous, I'm plotting this as a histogram where, once again, nonsense blue, synonymous green, but I'm also including the missense variance in gray on the reverse. So density being the y-axis, functional score being x-axis. And there is a somewhat bimodal distribution where nonsense are centered around 0, synonymous around 1, but there is overlap between the two. However, it is a minimal amount of overlap, which is nice to see. However, it is something to indicate that the data isn't perfect, despite it being of reasonable quality. And same thing on the flip side for the missense variance, it does seem to separate into a bimodal distribution where the majority of variants seen in the assay are tolerated with the density predominantly over 1, but there is a smaller subset of variants that receive damaging scores akin to nonsense variants. So that was some internal quality control. What we also, without further ado, will hopefully get into is the variant effect map itself. So make sure we're all situated. Blue is damaging, white is tolerated, red is hypercomplementing, yellow is the wild type, and gray is filtered or missing data. So spanning across amino acid positions x-axis, amino acid substitutions that we introduced on the y-axis. Um, so first off, one of the things that was actually somewhat surprising was that we did not seem to capture damaging variants in the nuclear localization signal highlighted here. And so this is somewhat surprising considering that nuclear to cytoplasmic localization of STK11 is a known component of activation. However, there are a couple potential explanations. Either one, the NLS is fairly robust to individual missense variations, or two, and I believe that this is the more likely case, simply put, the localization of STK11 is dispensable when it comes to this assay. What's most important is kinase activity. Is the kinase functioning rather than is the protein localized in the correct compartment? And also quickly mention, you can see on the bottom left here, the tolerated nonsense variance at the end terminus, stopping around position 22. Okay, so moving along the protein, we do recapitulate known features of STK11. So here are some known kinase motifs, first off being the GXG-XXG motif. So this is a glycine-rich loop that acts as a flexible clamp for ATP. And what you can see is that the variable position, the X's, tolerate nearly all substitutions, while the glycines, the invariable positions, seem to be more sensitive to hydrophobic substitutions compared to polar substitutions. The ATP binding site appears to be highly intolerant to variation, having nearly all substitutions be damaging. And similarly, a salt bridge located nearby, while not as critical as the ATP binding site, still does seem to be important for the protein's function. Okay, next, sliding along. Now, since this is a kinase, one of the most important sites is going to be the active site, the site of catalysis. And really, what I'd like to stress here is the position-dependent sensitivity to variation, where it should jump out that positions 176 and 181 are highly sensitive to any variation, while the other positions have intermediate or tolerated levels when it comes to substitutions. And so what I'd like to show you now is I've painted the crystal store, the functional scores onto the crystal structure and shown it in the 3D space. So here I'm zooming in into the active site specifically, and I've highlighted positions 176 and 181 to show that the side chains of these amino acids seem to be in close space in the 3D space, and so evidently the structural arrangements of their side chains are very important for catalysis. And therefore substituting to any other amino acid breaks this interaction and prevents the function of the kinase. So next, we also do see some other known features, some structural features associated with STK11. So here I'm highlighting two alpha helices that have been previously annotated. But once again, I'd like to stress the position-dependent sensitivity to variation. The alpha helix is annotated, for at least for this one, from 279 to 290, but clearly only a portion of this alpha helix is sensitive to variation. And so once again, painting functional scores onto the crystal structure, but here I'm showing the helical structures, and I've highlighted the different alpha helices that have been previously annotated. And what it seems to be the case is that positions that are internally focused, ones that are facing the active site, facing the binding partners, seem to be more sensitive to variation compared to externally facing residues. And this really is highlighted by the alpha helix at 260 to 269, which is external to the core of the protein and is entirely tolerated. While for the other alpha helices, there seems to be almost a periodic nature or really a position-dependent nature on sensitivity to variation. So now we get to the C-terminus of the protein, and here it really does seem, at least in this assay, that the vast majority of variants are tolerated. Now that's not to say that there aren't elements in the C-terminus that have been known to be important for STK11 function. Here I'm showing a membrane localization motif that seems to be dispensable in this assay. And so essentially, this raises a big question of if we're not capturing all aspects of STK11 function, are we accurately describing the disease-related characteristics of STK11, and is this assay going to be able to accurately classify pathogenic and benign? But before I get into that, I'd first like to talk about some alternative functional data that we were able to get access from collaborators. And so here, first off, I'll talk about some data from Dr. Dave Adams' group from the Wellcome Sanger Institute. So x-axis is our HeLa-based functional score, and on y-axis is data from their HAP1 essentiality assay. So what they saw is that knockout of STK11 in HAP1 cells has the reverse phenotype to what we're seeing here, where HAP1 is essential, where STK11 is essential in HAP1 cells, and so variants that affect STK11 function will drop out of their population. But regardless of the specific experimental details, it does seem that for the most part we are in agreement where stop codons are damaging the bottom left for both assays. Synonymous variants are tolerated for the most part in the top right in both assays, and missense variants seem to have a bit of a mixed distribution between the two. Now, of course, I should still mention that there are variants in the top left and the bottom right that are potentially misclassified. However, there could be more biological reasonings. For example, the HAP1 experiment was performed with endogenous locus. The HeLa-based experiment was a cDNA-based experiment, so there is the potential for splicing effects to be the reason behind the differences between the assays. But overall, there is a moderately strong Pearson correlation of 0.56 that is highly significant, and these assays do show reasonably good concordance between the results. We're also lucky enough to have collaborators at Tango Therapeutics who performed a slightly more complicated system where they actually worked with a mouse model and performed a xenograft of A549 lung cancer cells. And so there is potential rationale that a more complicated model might be able to capture elements of STK11 function or of protein function that a more simple model might not be able to capture. And so this could be the C-terminal regulatory events that we're not seeing in the current assay I've described today. But once again, there does seem to be fairly good agreement for the most part between assays where, in this case, I've broken it down into kinase domain or the N-terminal to C-terminal components. And it seems that pretty much all variants that were found to be damaging in both assays were found to be in the kinase domain, which makes sense considering the data that I just previously showed you. But there are a subset of variants in the C-terminal domain, shown in pink here, that were tolerated in the HeLa-based assay that I presented today, but seem to have mildly detrimental effects in the Tango xenograft assay. Not akin to a completely dead kinase variant, but there is potential that there is a mild effect for certain variants that is getting picked up in the xenograft assay that we are not seeing. But once again, overall, there does seem to be a fairly reasonable Pearson correlation of 0.59, highly significant p-value, and there is reasonable concordance between the two experiments together. Now, we also do have other alternative data that was lower throughput. So here, this is previously published data by Dr. David Seward at the University of Vermont. Essentially, what they've done is they looked at p53 expression, so a luciferase-based p53 assay, as STK11 is known to interact and affect the expression level. And so, from a completely independent low throughput assay, we are showing perfect agreement where variants that are damaging in the proliferation assay seem to have lower levels of luciferase activity compared to variants that are tolerated in the assay, which have higher levels of luciferase activity. And so this is a highly correlated data set, 0.92, that is significant. And while it is a lower throughput, while it is a lower number of total variants, it is nice to see that we do recapitulate completely independent experiments that have independent readouts from the one we are performing here. And similarly, and sort of to touch on the nuclear localization element, we collaborators at Dr. Taipali's lab performed a GFP STK11 fusion experiment where they used microscopy to then look at the subcellular localization of STK11. And so on the y-axis, I'm plotting the intensity of GFP from the nuclear to cytoplasmic compartments. And essentially, what we're seeing is that variants that were damaging in the proliferation assay seem to be sequestered in the nucleus. They seem to have a high nuclear to cytoplasmic ratio and therefore are predominantly in the nucleus, while variants that were tolerated in the assay seem to still be slightly elevated in the nucleus, but have a much more equivalent distribution between nuclear to cytoplasmic components. And once again, this is a smaller number of data points, of course, but the correlation is fairly high at 0.94, and it is significant as well. So essentially, just to summarize the past two slides, we do have a reasonable amount of alternative functional data that has been produced, both high throughput and low throughput. And overall, we do seem to show fairly good agreement, both in terms of Pearson correlation, as well as just simply looking at concordance across the midpoints with all of these assays. So now, and this is really probably one of the more important slides, is how do we do at classifying annotated pathogenic and annotated benign variants? So we collaborated with Ambry Genetics, who provided us both with the reference set shown here, as well as then performed a variant recuration of effort to incorporate the functional data with external sources of evidence. And really, what I'm really happy to say is that, yeah, this assay, despite not being able to capture certain localization elements, does seem to accurately call every single variant in this reference list. So all 11, all 74 benign were accurately classified by this assay, as indicated by this precision recall curve, and the area under the curve being 1.0. So that's, of course, very reassuring for us. It is nice to see that we are, in fact, relevant to the real world patient individuals. And so what we've done is then we tried to translate the scores we have to the ACMG AMP evidence strength categories. And so what I'm showing here on the right is log likelihood ratio calculations, where what we've done is we've taken the reference variance from the left, the pathogenic benign, plotted them as the vertical blue and green lines on the functional score range shown here on the x-axis, and then calculated probability density functions for each of them and looked at the ratio between the two. And so because we have such good clean separation between pathogenic and benign, we are potentially able to ascribe up to strong evidence of pathogenicity and strong evidence of benignity depending on your functional score. So if you have a functional score of 0 or below, then yes, maybe this would be up to strong level of evidence for pathogenicity, and 1 or above, maybe potentially strong evidence towards benignity. And so using an internal list of variants of uncertain significance from Ambry Genetics, and then also incorporating with external sources of evidence, we are potentially able to reclassify nearly or over half of the 516 variants of uncertain significance that they had in their database towards benign or pathogenic, with the vast majority going towards benignity, 295 to benignity, 8 towards pathogenicity, and 213 that did not have sufficient evidence to be reclassified or to potentially be reclassified to a more informative designation. So this, of course, is really potentially has is potentially able to address that variant interpretation crisis that we showed in the first slide, as this is fairly high throughput. It is testing thousands of variants, and as more and more data sets and more evidence is provided, we'll hopefully be able to really address this at scale. So finally, I'll leave you off on one or two more slides, just looking at integrating with clinical phenotypes. So here, what I've done is I've partnered with the Zane Cohen Center, which is a gastrointestinal clinic, and essentially I did a retrospective both patient chart review as well as online literature review to collect data associated with the age of onset of Peutz-Jeghers syndrome on the left here, as well as the age of onset of cancer for individuals that have PJS on the right here. So pretty much, once again, small sample size. There is variability in the data as I collected this from multiple publications, multiple patient charts. But generally speaking, it does appear that damaging variants appear patients with damaging variants appear in clinic earlier in life, where we can see that below the age of 10, there are individuals that are presenting with damaging variants, while individuals that have tolerated variants appear to present slightly later in life, maybe 15 years of age or above, and even potentially some incidental findings much later in life, ages of 40 plus, indicating that these individuals did not have severe clinical phenotypes and therefore were not being assessed for Peutz-Jeghers syndrome. But once again, this is a small data set. There is variability associated with it. And so while there is the potential indicator that there is distinction between the two, the Pearson correlation is moderate with a significant but not very significant p-value. And same thing with the age of cancer onset for individuals with PJS. This is a very small data set, but it does appear that the individuals with damaging variants might present with cancer ages around 40, while individuals with tolerated variants might present with cancer around the age of 60. And so while there is a high Pearson correlation, it is somewhat low significance due to the very few number of data points tested. But really, this is essentially potentially indicating that we can stratify patients who are in more need of close monitoring, close care, compared to individuals who have a tolerated variant and might have a better outcome and course of disease. But okay, now this last slide really, I think, will hopefully drive this home for somatic cancers. So what I've done here is I've collected publicly available data from cBioportal, and I'm splitting into both pan cancer and as well as non-small cell lung cancer focus specifically, because lung cancer being one of the most predominant STK11 tissue types that are affected. And I'm also separated into variants that were damaging the assay, variants that were tolerated in the assay, and and individuals that did not have an STK11 variant. So essentially like a negative control. And what I'm showing here is a Kaplan-Meier survival curve where we have years post-sequencing on the x-axis and the probability of survival on the y-axis. And so what hopefully should jump out is that the tolerated variants, individuals with a tolerated variant seem to have better outcomes compared to individuals with damaging variants, where if we look at the 50% survival probability, this could be the difference between 2 years and 4 years when it comes to overall survival, which is dramatic. And if I do log rank tests to look at if they're statistically distinguishable from each other, tolerated variants are statistically indistinguishable from no variant controls, while individuals with damaging variants do have worse outcomes. And this is reproducible both in an independent data set that I'm not showing here today, but also if I specifically look at lung cancer, the trend does remain. And so to circle back to one of the earlier slides, essentially, if we are able to proactively assess the genotype of a lung cancer patient, and it's found that they have a damaging STK11 variant, then perhaps they should not be provided with an immunotherapy and should instead be provided with a different, more effective therapeutic. Now, this, of course, is not breaking it down. The data I'm showing here is not breaking it down by treatment type, and so there is, of course, more evidence that needs to be provided. But generally speaking, the functional data I've presented today is effective at stratifying pathogenic variants from benign variants. And so with that, I'd just like to thank, of course, everyone involved in the Roth Lab, as well as our various collaborators. And then thanks for the organizing committee for inviting me to chat today, and happy to answer any questions, of course.
Great. Thank you for our speaker, Daniel. We have a few comments on the Q&A. First from Sharon. So this is early in the data presentation. The synonymous variants can cause splicing. So the overlap, I think, between the two modes you're showing is the overlap doesn't mean that it isn't perfect.
What I'll say is that if this were at the endogenous locus, I would absolutely agree. But because this is a cDNA-based system where we're using a landing pad to stably integrate the cells, we're not expecting to see any effects of splicing. And so the intermediate variants, or the detrimental synonymous variants here, it's unlikely to be due to splicing.
Okay, great. We have a technical question from Mariano. How did you introduce the library into the HeLa cells? Did you use lentiviral transduction or a landing pad system?
Yeah, so, and here, maybe I can skip all the way to the end. Essentially what we've done, oops, sorry about that. Essentially what we did is we used a landing pad system, and here's just a rough overview of it. Pretty much there are recombination sites that have been engineered into the AAVS1 locus. And when we transfect a plasmid as well as the BXB1 integrase, what happens is a single recombination event occurs, which obliterates or changes the recombination site, the AppB site, and allows for a single stable integration event at the AAVS1 locus.
Great. We have a few more.
Mhm.
So this is from Chen. Is log likelihood same as all-pass score? I think this one is second to the last slide.
Yeah, so this is what I'll say it's very similar in the sense that we are also calculating essentially what is an odds pass score, but with the log likelihood ratio, what we've done here is we've broken it down into bins. So this is sort of a moving window across the functional score where, essentially we're saying that if you have a score of zero point 0.4 or zero point 0.3, maybe we're not as confident about providing evidence as a score of 0. So it is very akin to an odds pass score, just sort of in a moving window analysis.
Okay. One more comment from Sharon
Oh yes.
about the age. So he or she is saying that as a clinician, I will not discuss the age of diagnosis data with a lot more examples. Remember that many individuals are diagnosed with PJS due to the pigmentation that shows up around age 4 to 7. Perhaps some of your Missense score as pathogenic but don't have the typical pigmentation.
hmm Yeah, of course, that's a great point to make. Like, I am very hesitant to talk about the Putz stuff just because it is a very small sample size. But I think that sort of when we're working with rare disease, it is so hard to really be able to get that statistical power to be able to make confident statements. And so when it comes to sort of the Putz charts that I was showing, yes, this is by no means definitive. I'm not breaking it down by the reasoning or the indicator of diagnosis. This is sort of meant to be a sort of a broad top level view of it. And honestly, it would be amazing if the data was available to dig deeper into sort of specific clinical phenotypes. So things like how many intestinal polyps do they develop? Essentially like if the data was available, I would love to dig in further and hopefully be able to give a better understanding of how the functional scores relate to Peutz-Jeghers syndrome. But just with the data I have available, I can make sort of hopefully trend statements, but nothing definitive at all, because of course there are a lot of confounding factors. Most notably patient-specific variability, as the same variant, the same individual missense variant might have different clinical outcomes depending on the patient.